Hi, everyone. Welcome to our first ever DSAI event, a lunch and learn. So my name is Kate Taylor. I'm an assistant professor in the biology department. And I want to start by thanking Paul for organizing this series. And my understanding is that the goal of this series is for people in different disciplines to talk about the computational tools that we're using and their applications to hopefully learn from each other. And so I'm going to talk a little bit today about my work looking at genomics and why the tool of BASH is so important for being able to solve problems in my field. So I'm going to start with a short introduction, kind of giving you the motivation of why you might want to learn this tool. And then we're going to kind of take a pause. We're going to all get set up accessing the Hofstra computational cluster, and then you're going to get to try using this tool, kind of coding along with me as kind of the second half of this event. Okay, so the reason that I use BASH is because I'm really interested in understanding genetics. And so I think many of you are familiar with the idea that all living things are made up of cells. And in those cells, there's genetic information. And DNA is the chemical compound that contains the information that's needed to develop life and to direct the activities of life. So each DNA strand is made up of 4 chemical units that are called nucleotides or bases. And they're kind of like a genetic alphabet. The bases are adenine, thymine, guanine, and cytosine, and they're referred to as AGTC. You're probably also familiar with the idea that genetic information is passed from parent to offspring, and that all of the genetic information that an organism possesses, that complete set of DNA, is called a genome. You might not know that one copy of the human genome has about over 3 billion base pairs in it. So if you wanted to know all the genetic information in the human genome, it would be 3 billion A's, G's, T's, C's, one after another, which is a lot of information. And the genomes that different people have and different organisms have are more similar than they are different. The average 2 people, their genomes will be less than, they'll be 99% similar. So less than 1% of the genome will vary between individuals. But those differences among individuals that are called genetic variants can be very important. And this is what makes people unique. And by comparing the genome sequences of different people or different organisms, we can get insight to the impacts of these variants. And so a person's set of genomic variants is what makes them unique. And this information is really, really important. It's important for understanding fundamentals about human biology and genetics, but this can also be used for really exciting approaches in personalized medicine, like targeted treatments for different cancers or diseases, for predicting genetic disease risk, for genetic carrier screening, and even for applications like forensic ancestry testing, much, much more. There are lots of reasons why you might want to know the genetic information that a human possesses. But to know a genome sequence is not an easy task. So, DNA, it's this super tiny structure that's inside of our cells. We can't just read it. It requires molecular techniques and really specialized equipment to be able to get that information out. So if you wanted to know a genome sequence, you have to start by getting the DNA out of the cell and purifying it. You then have to prepare the DNA for sequencing. And the reason you have to do this is because we don't have perfect technologies, right? We can't just say, magic technology, tell me what all of the nucleotides are that make up a human's genome. So with the technological constraints of today, you need to break that genome into small pieces, like usually about a couple hundred base pairs long, just a couple hundred nucleotides. And then often you're going to need to do some amplification protocol, which is going to be a little bit random, resulting in some parts of the genome being more and other parts less represented. So then you put your DNA onto a sequencing machine, and then to simplify, you're going to generate many, many sequencing reads. And the reason that you need many, many sequencing reads is, well, we said 3 billion base pairs, right? And each read is only a couple hundred base pairs. But we can't just sequence every base pair one time. Technologies for sequencing are imperfect. Sequencing errors occur. And so to know any one individual's, genotype at a specific position in the genome, you're going to need to sequence that region multiple times to have confidence that your result is not an error of the sequencing machine, but real and biological. So you end up sequencing regions multiple times, maybe 10s or 20 times, just to be confident that sequencing errors aren't underlying what might seem to be a genetic variant. And then also sequencing is not evenly distributed, meaning that you need to sequence more to capture all of the regions. And what this means is that you need to get a lot of data, right? We're saying there's 3 billion base pairs, and then we need to sequence all of that multiple times to correct for errors. And we're going to need to sequence it deeply to be confident we've gotten all of those base pairs. And then you can start to assemble those reads computationally and then start to infer the sequence of the genome. And so this is what a lot of my work is interested in, is kind of these computational approaches to work with this data and extract genetic information. So if you sequence an organism's genome, like what happens? What does that data look like? Well, the data that you'll get off of a sequencer is usually a plain text file. And that plain text file is called a FASTQ file. It'll usually have an extension that's .fastq. And it contains information about sequence and also quality. And it has this prescribed format where there's a first line that gives you information about, like, what did the sequence come from? Then there's a second line that has the nucleotide sequence. So this would be, for our application, the couple hundred base pairs that were that single read. And we're going to have many, many, many of those reads for our full sequencing run. And then a plus. And then the next line has encoded nucleotide quality scores, where each symbol represents confidence in that base call. And there's a way you could translate these symbols into percent confidence that this base call is true. Yeah, it does. Yes, and we are very happy to have great computer programs for interpreting this data, because it would be very difficult for me to look at this data and be confident and understand how reliable this data is. Yeah.
Which symbol is the highest quality?
Oh, what's that? What is the highest quality? I don't know off the top of my head.
Hopefully percent greater than, because that's the one you got the most in.
This is one of, so we're going to look at 2 example files today, and one of them is kind of high quality and one of them is low quality. And this screenshot is from the high quality one. So I think this is pretty good quality. But you can see that is not a very compact format for storing data, right? We've got multiple, we got multiple representation of each part of the genome for all of the multiple times we sequenced it. Eventually, this data gets stored in a more kind of compact FASTA format, which removes the quality information after you're confident in your base calls, and will usually give you the resolved sequence rather than the raw data. Okay. And so if you took that FASTA file and you printed it out, it would be huge. So someone did it. They printed it. Yes, this is one of the first human reference genomes printed out. I think that each of the colors on the spines of the books represent different chromosomes. So this is the resolved sequence for a single human genome, and you could see that, like, it's much taller than this guy, right?
Waste of paper.
So this is big data, right? And the data is just getting bigger because sequencing technologies are advancing quickly. And so in 2001, which is when the first human reference genome was published, the first human genome was published, it cost about $100,000 to sequence a genome. And as technologies are improving, you can now sequence a whole human genome for less than $1,000. And I think it's generally thought that the $100 genome is within reach, which is super exciting, right? This is great news for all of those applications we talked about. but it also means that genomics data is just huge. And so genomics data is expected to generate 40 exabytes of data in the next decade. And I mean, exabytes, like what is that even? Like that is just so big, it's almost impossible to think about for me. So I think this graphic is a little bit helpful. So if each shark here is 100 million gigabytes of data, then all of the sharks are 40 exabytes of data. And so we're like adrift in an ocean of big data, right? And so from this, I think you can probably tell that genomics data is almost always too much for your laptop or your personal computer to handle. And you're going to really need a large computer or a cluster of computers to start to gain insights from this data. These are often called remote computers, high performance computers, or computing clusters. But to interact with these tools, with these high performance computers, you need to be able to speak their language. And the language that they use is called Bash. And so today we're going to start kind of playing around with some genomics files using some simple Bash commands to get started. And I think that probably most of you are not biologists, but Bash is just a very widely used tool. And that if you have any kind of big data problems in your field, I think that this can be really, really helpful for you as kind of your way to start to access high performance computing resources. Yeah.
Is this distinct from Bash shell in Unix or are they related?
It is the exact same thing. So Bash is the most commonly used shell. And so that is what we'll be talking about today. Yeah. Okay, so let's get started. You guys don't just want to hear me talk for a full hour and a half. And so to get started, I want you guys to be able to follow along with what I'm doing, to use your own computer and start typing Bash commands. And so to do that, you are going to access our Star HPC high performance computing cluster here at Hofstra. And so you should follow these instructions. You're going to navigate to this web address. You're going to click login with a local account. I believe that Dr. Yang distributed to all of you a piece of paper that has your username and password. I'm going to type in those credentials. And then you're going to click Start Shell Access. And then it's going to say like, oh, are you sure you want to connect? And then type yes. Okay, so we're going to take a pause for 5 minutes or 3 minutes while everybody gets started. And if you're having trouble and you're stuck, raise your hand and I'll try and come around and help you troubleshoot what's going wrong. Or maybe it looks like Michael also looks like ready to jump up. So I think our IT wizard might also be coming around to help you. They're already available. Yeah.
Yeah, turn on the link.
Oh, yeah.
There's an icon on the right side of the box.
I don't have.
Oh, yeah, that is tough. I think I copy pasted mine, but that is a long string of symbols and letters.
So it's the only data that FastQ.
Yeah, the only files we're going to use are those two FastQ files. Yes.
I will send it on Zoom.
Oh, thank you for doing that.
Okay, that way people can at least play with it.
But I think that if the people on Zoom are on the cluster,
If they already have cluster network account, they should work. But if they don't,
then they can at least look at the files.
at least they can look at the file.
Yes, yes.
They have Unix.
Yeah, if you have Unix, you should be able to just
So
do it.
you can tell them if they have Apple open.
Yes.
Sometimes they may not be aware of it.
Okay, I'm walking around, hoping that if somebody is like totally stuck and unable to open up that shall send them that they're gonna raise their hand or shout out to me.
The PowerShell has a couple commands.
Yeah, it's Windows
Oh, it
commands
doesn't have
though,
breadth,
it's
so-
horrible.
It doesn't have breadth. No.
If you're on Windows, I'm working with WSL, but that's a whole
Yeah.
different story.
Well- I was gonna ask you about this stuff.
Yeah, get WSL.
Eight,
No, it's free.
like searching, and there's this one for $10.
Just do something. It's free.
Yes.
Okay, and if you're still working on that, so here were the instructions again. Are people generally, are we on? Are we in? It's yes, thumbs up. We're going, we're good. Okay, that's great.
Get timed out if you're not active.
You do get timed out if you're not active.
Just like Microsoft.
Password is a big challenge to do.
Okay, all right, so I think most of us are probably more familiar with interacting with computers using what's called a graphical user interface, right? That's when you can use your mouse and you're clicking buttons, and that makes things very simple. But to be able to access remote computing resources, you're going to have to work on a command line interface. And there are a lot of good reasons to do that. And one of them is to, access remote computers. But also for a lot of field-specific tools, at least in my field, and I'm guessing in your field as well, there might not be a graphical user interface available. You might only be able to use that tool or program on the command line. And also working in this way gives you a lot of power to work efficiently. Tasks that could be really repetitive using a graphical user interface, like renaming files, can be super quick and easy using the command line. And so it can hopefully automate tasks that are tedious and allow you to be more productive. So if you have a Mac or a Linux machine, you can always access the shell and the bash shell using a program called Terminal. And that way you can interact directly with your personal computer. What we've launched here today is a way to interact with the remote computing cluster, the Star high-performance computing cluster. If you have a Windows machine, you're going to need to download a separate program to access a shell on your computer, which is part of why for today we're all going to be working on the cluster together. So we're all in the same environment, and also the data and software that we need are all going to be there and are going to be installed. Okay, so I want to start by talking about navigating the file system on our computers. Because in order to do almost anything, you need to be able to point tools towards, here's the file that I want you to run this analysis on. Here's where I want you to put the output of that analysis. And so all computers, they're organized into this like directory structure, right? They're made up of different folders. On your home computer, it might be like, oh, you have a desktop folder and a downloads folder. And so you're going to need to know the structure of the computer you're working on to be able to use it efficiently. So we're going to explore and navigate this file system as kind of our first start in Bash. But you can see that I got timed out because I was too slow. So I'm going to close this. The timeout is pretty aggressive. But let's try again. Okay. So okay, so what you can see here is that at the bottom of your screen, you have what's called the command prompt. So this is listing my username, and you can see this $sign. So this $sign is the prompt, and this lets you know that Bash is ready to start listening to the commands that you're going to pass it. So we can try typing lots of different commands here. The first one that I want to use is called pwd. And so this is going to print our working directory. So pwd stands for print working directory. And for all of you, you should see that you're now in a folder within another folder. So there you can see it prints out a directory structure. So this first backslash, this is the root. And then from there, you can see there's a folder that's called home. And then within the home folder, I'm in user 002. That's the folder, that's my home directory. Your home directory is going to be slightly different. It's probably like user 017 or something like that. So that's the folder where we are now. But in order to do this tutorial, we're going to need to retrieve some files to start to analyze. And so to do that, we're going to need to move around the cluster directory structure. So right now we're in our home directory. We're going to come back here and we're going to work in our home directory. But first, oh, I'm very sorry. Uh-oh.
You might tell the sysadmin to expand that.
Yes. It is very short. I'm so sorry. This is our very first one of these workshops, and we are going to be working out the kinks this semester. Okay, so we're going to be moving around our file system to retrieve the files that we need for this tutorial. And the way we're going to do that is using a command called cd, which means change directories. So the command is cd, and then you have to tell the computer where to go. You have to give it like really clear instructions about how to move in the file system. And so I'm going to show you how to find the files that we need, and then you're going to tell the computer how to find it too. So we're going to start at the root, and the first folder that we're going to look in is called fs1, and then dash shared, dash, and then we're going to say lunch and learn, but that'd be a lot to type out. So you can try typing, if you tab after you start typing lunch and learn. So I typed like LUN, and then I typed tab, and then it says, oh, okay, you want to go to the lunch and learn folder. And then we're going to go to September, whoop, spelled correctly, September 24th. And again, I used tab completion, so I just typed S, and it filled in for me that I wanted the September 24th folder. And then a folder called FastQ. So the instructions are telling the computer to move through all of these directories. So if you manage to do that without typos, then it should have quietly moved. And then the only change that you're going to see is that before your command prompt, you'll see the current folder that you're in. Okay, so we're in this folder that's called FastQ. We can type pwd to confirm that it worked. And yes, we're in the folder we tried to get to. And then let's look at what's in this folder. Let's see how it's organized. So you can see all of the files and directories or folders within that folder using the ls command. So ls stands for list, and it prints out the names of files and folders. And so you can see that if you type ls, you get 2 file names. They both start with srr, which is a kind of a biological field thing that this is, that it's coming from a sequencing read archive. And they end in .fastq. So we've got 2 files there. So if you want to move around and not need to memorize the full path to the place that you're going, there are some special directory names that can be really useful. So one of them is the single dot or a single period, which refers to the directory you're currently in. And another is the double dot, which refers to like one folder structure above where you are. So right now we did PWD, we're in this FastQ folder. So if I use so if I use the double dot, then we should move up to the September 2024 folder. I'm very sorry for these difficulties, my friends, and I appreciate you bearing with me. So I'm going to repeat what I just did. So I'm going to CD into FS1. And then I'm going to use tab completion just to get into the correct folder. Okay, so we can try using that reserved directory shortcut, the double dot, to move around our system. So we can say CD and then double dot to move into the September 24th folder. And you can see that that worked because now before a command prompt, you can see the folder is September 24th. You can then you could list what's in this folder. And you can see that it has the FASTQ folder within it. And the color here indicates that it's a directory rather than a file. And I think that a lot of different terminals that you work with or ways to interact will similarly give you a different color for a file versus a folder. So because we have a folder, we can CD back into FASTQ. We can LS, and we've still got those two files there. So you might be interested in starting to automate something. And with two files, it's not that hard to just do things twice. But if you start having more files, it becomes really important to kind of automate tasks across multiple files. And a really useful way to do that is using what are called wildcards, which are the asterisk character. So it can be used to represent any character and any number of characters. So if we wanted to do something to all of the FASTQ files in this directory, for example, like if we just wanted to list FASTQ files and not other kinds of files, we could type our ls command, and then we can use a wildcard. So this means anything before the period FASTQ. So this is going to match anything before with a FASTQ extension. And it still lists both of those files. But if instead we typed ls and then we said we want anything that ends with 997.fastq. Whoop. What have I done wrong? Oh, yep. ls anything dot 977 dot fastq. Thank you for catching my typo. Oh, and I didn't want the period. Oh, I just instinctually did something that I'll now tell you what I did. If you type something wrong and you want to pull back up what you just did, you can use the up arrow. You can use the up key. And so I just use the up to pull up what I typed before. I'm deleting the extra period that I put in. And then now, because I've removed all of my typos, you can see that there's this one file that matches this. Okay, so if we want to start to learn more about our data, we want to be able to look at these files. And a really simple way to start looking at files is using the head and tail commands, which respectively let you look at the beginning and ends of text files. So we can type the command head, and then we can say that we want to look at SRR and then 097. And I'm going to use tab completion, so I don't need to memorize that long list of numbers. Okay. Okay, so I've typed head, and what I've gotten is a FASTQ file. So earlier I said that, oh, we've got this FASTQ format. The first line starts with an @ and that tells you where the sequence came from. The next line is going to be the sequence itself. So that's your A's, T's, G's, and C's. The line after that is either just a plus or a plus in the repeated header. And then you have quality score, encoded quality scores as the final line. So if you didn't want to print out, the predefined number of lines that head wants to do as a default, you can provide more arguments to that command to change its behavior. So when I type head by default, I think it's printing out 10 lines. But if I just wanted it to do something different, I can provide additional arguments. Oh no, I've got to start talking faster. You can provide additional arguments to modify the behavior of these tools. Okay, sorry. So I'm going to go back and I'm going to go back to FS1. You guys must be really annoyed because I bet you're doing the same thing over and over again if you're actually following along. I'm very sorry. We're just
Keep typing LF.
Skip the.
Oh, that's a good idea. A little LS every once in a while. Okay. Yes, that is very smart. Okay, I will start doing that. Mm, yes, that is a good point. Okay, so we wanted to modify the default behavior of the head command. So we're going to type head and then SRR 09, and then I'm going to use tab completion. (097). And then we can specify an additional argument to modify this tool's behavior by putting a dash and then the name of that argument. And for the head command, a really commonly used option that you might want to do is change the number of lines that are printed by default. And so we can specify the number of lines we wanted after n. So if I want one line, I'll say -n1. And then that changed the behavior of the tool. If we wanted to just look at the FASTQ file for the first single sequencing read, we could type just what we did before, and I use the up arrow to pull up what I just typed, and then say we want N4. And so this gives us just the results for that first sequencing read. Okay, so we've started to look at these files, but to really work with them, I think it's going to be useful to put them in our own home directories. So in order to do that, we are going to copy them. So we can copy them using the command cp. And then we're going to use that wildcard again. So we're not going to need to copy each file. We can say cp and then our wildcard.fastq. And so this is going to copy anything that ends with .fastq that's in our current folder that we're working in. And then we're going to want to copy it to our home. We could type out the full path to our home, but there's a special reserved character for your home that's a tilde. It's this little one that's at the top of your keyboard. And that's a shortcut that's going to say, send it to home. I'm going to run that. It looks like nothing happened, but if we cd into home, and then we ls to see what's there, you can see that now I have copies of these two files. So there are lots of specialized tools for working with sequencing files, but I'm going to teach you a general bash tool that I think might be useful for working with any kind of text data that you might have. And that is so to start to explore these files, we can search for specific strings we might be interested in. Maybe we want to know, where does AGTC occur in a file? And so we can type the command grep, and then we type a string that we want to search for. So ACGT. And then we type the file that we want to search in. So it's going to look for any time ACGT appears in our file. So SRR 097, and then I'm going to use my tab completion. So this is searching for ACGT in that file. And I get this printout that shows me all of the lines in my file that have ACGT. Well, that is somewhat useful, but a little bit hard to parse. Maybe your question is, how often does that occur in my file? If that's your question, then you can add the -c argument to have it count the lines rather than print the lines. So we can say grep ACGT SRR 097, tab completing. Oh, and I forgot to do the -C that I was planning to do. And so then it tells me, okay, this appears 30 times in your file. So it seems like in the sequences we generated, that string appears 30 times. But that might not be particularly interesting to us. Like maybe a more important question would be, how often do we have unknown bases in our sequencing files? Where do we have really poor quality data where we can't figure out what the nucleotides are? So we could instead grep for strings of ends, which is how unknown nucleotides are recorded in these files. So we can grep, and then I'm going to type 10 ends, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And I'm going to look in SRR 097, and then I tab completed. And it does not find it in that file. But what if we look in our other file? So we're going to this time look at SRR, let's see, 098. Okay, well, this one, we've got a lot of ends. So let's switch to counting rather than printing every single one of these. So we can switch to counting, again, using the C argument. There's 134 sequences that have lots of ends in them with all of this unknown data. But you know, it might be useful to just print the line itself, but because this FASTQ format has lots of information about those reads recorded in the lines above and below, we might want to extract more of the lines from the file. And we can do that using additional arguments. So I'm going to use the up key to bring up my last command, and I'm going to add some additional arguments. So they're called B and A. So it's before and after. So -B, you say how many lines before you want. And so I know that one line before the sequence is going to be the identifier. So I'm going to want one line above. And then I'm going to say -A after. I want 2 lines because I've got that plus line and then I've got the line with the quality scores. Okay, so I want to look for this string, and then I want to print out the line 1 above and 2 below. Oh, but I need to remove my C. You can see that I often takes me multiple attempts to get a command working properly. Okay, so now we've not just pulled out the lines that have the nucleotide sequence, but the proper FASTQ format for those reads. And so maybe we'd want to save those reads or do something with them. And the way that we're going to do that today is that we're going to try redirecting the output. So we can take, we're going to use the same exact command, so we can use the up on our keyboard. And we're going to use a greater than symbol. And so a greater than is a redirect. And so this is telling the computer, I want you to save the outputs of this command rather than just printing them to the terminal, save them somewhere else. And we're going to specify that we want to save them in a file called badreads.txt. Okay, so again, it seems like it might have worked. We didn't get an output because we've redirected our output. It's no longer being printed in the terminal. It should have been saved as a file. And we can use ls to check and see if badreads.txt exists. And it does now, which is very exciting. So we can, again, use head just to confirm that badreads.txt has the output we expected, which is our bad reads that are full of lots of ends. Okay, so this was kind of a short way to kind of dip our toes into using Bash and kind of show you a couple tools and some skills for starting to work on the computing cluster. But we're going to be having more sessions later this semester, including one that's going to be fully focused just on using the computing cluster, and I think is going to get us to a little bit of a higher level than my intro here. But I hope that this has been interesting and that maybe you've learned a little bit about genomics or BASH, and I'd be really happy to take any questions that you guys have.
Thank you, Kate.
Yeah.